All right, so if you are signing on just now, we're going to get started in a minute. And if you're interested in any of our services or Spectral Flow, you can check us out on taco.bio and just click products and services and look for the Spectral Flow webpage. So look forward to seeing everybody shortly. Okay, so we're going to get started. My name is Ramji Srinivasan. I'm co-founder and CEO of TACO, and our mission is to make cytometry simple for clinical trials. And today we're going to talk about our 25 marker spectral flow validation on fixed whole blood. Specifically, we're going to cover inter-run and intra-run reproducibility performance. This is a major hot topic for drug developers, and we'll see why. So let's talk about the problem that drug developers face. And in short, it's a challenge of which sample or specimen to collect on trial. So for drug developers, when collecting blood for immunophenotyping, a lot of times people have turned to PBMCs or peripheral blood mononuclear cells. The issue with these is they're very expensive to collect. They require specialized equipment. They miss granulocytes, which have a key impact on the immune system. And the viability is all over the place, anywhere from 13% to sometimes you get great viability, but that varies from collection to collection. The other alternative is live specimens, which have a 10 to 20% failure rate. So imagine if you're paying for 100 specimens and 10 to 20% don't even get a readout on the instrument. As a result, people have tried to get fixed samples to work, and with fixed samples, you can snapshot the cells in place. The challenge is fixation isn't yet optimized for high dimensional cytometry. And that's what we set out to do with our TOCO kit, and we're going to talk a little bit about that. So why fix blood at all instead of running fresh? So what does snapshotting achieve? Well, the first is this 10 to 20% reduction in failure rate through the centralization of flow analysis. You don't need a physical site in every geography, from Edinburgh to South Africa to Japan, etc. Second is you can batch samples. So you can store a set of samples until a whole set, i.e. all the time points from a patient are complete. And third is you can reduce your committed processing costs and expand your analysis window. So if you don't know which samples to analyze, no sweat. Just store the samples at -80 until you're ready to decide which samples to analyze. So you can actually bring down your fees because you don't have to run every sample off the machine. So let's talk about our method for this experiment. First, we use our TokuKit collection process. And if you are interested in TokuKit, you can check it out at taco.bio/TokuKit. First thing is you just draw into a green top or purple top tube. So you invert that 10 times, it's 4 ml. From this tube, you take 2 mLs of blood and put this into buffer 1. Buffer 1, you invert 10 times and then keep that stable for 15 minutes, all at room temperature. And then you take this entire concoction, take 2 mLs out of that and put this into buffer 2, invert that 10 times, and then you can store on -80, or basically ship to us on dry ice, and we can store at -80, stable for up to 24 months. So this is the collection process that we followed. Here's the spectral marker panel. So we did 25 markers, everything from CD66B all the way down to gamma delta TCR, and you can see the fluorophores listed here. This panel allowed us to look at 136 immune subtypes across 36 cell populations and 6 functional markers. So you can see over here, starting at leukocyte, going all the way down to, say, CD4 T naive, CD8 TEMRA, type 1 conventional dendritic cells. And this little dot over here, this little number, is the number of functional markers we looked at in that particular cell type. So, for example, PD1, we looked at T cell, B cell, NK cell monocyte, and dendritic cells. Okay. Then we had Toku kit and the panel meet. So we have fixed whole blood from 3 healthy human donors. So here are the 3 healthy human donors over here. We did our fixation at TACO, and then we ran samples on a 25 marker Aurora spectral flow panel. So for each blood specimen, we had a 25 marker immune cell panel that we just talked about. We started at CD3 all the way down to CD56. And then you can see the rows here are all the cells that we acquired. So hundreds of thousands of individual rows here. And now you got this massive matrix, 25 by 1.4 million. And you got an individual data point in each one of these cells of the matrix. So the drug developer's question is, how reproducible is this thing? That is, if you measure this million point matrix each time, are you going to see approximately the same thing each time? And you can imagine if you step on a weight scale and one day you're 50 pounds, next day you're 100 pounds, next day you're 200 pounds, that's not a very reliable weight scale. So the drug developer's task is to figure out, are tests like ours reliable for measuring immune cell activation during their drug treatment? So let's go into some of the reproducibility tests that we administered. In our validation plan, we had 2 measures, intra-run precision and inter-run precision. So intra-run, same sample, same run. Inter-run, same sample, different run. And the criteria is a coefficient of variation less than or equal to 20% for greater than 95% of populations. All coefficient of variation means a standard deviation divided by mean. So the higher the number this is, the worse, the lower the better, because it doesn't vary that much. And we looked at 32 populations per donor in these. So let's go to intra-run. Intra-run is taking the same sample, aliquoting it out, and running these all, basically doing the fixation, running these all through the cytometer, acquiring the data, and comparing these. So same sample, same run, probably the most basic of tests. And for this, we found great CVs here, 4.76 across all 3 donors. So if you look over here on the x-axis, this is the median size of populations. And the y-axis is the percent CV of percent of non-granulocytes, so higher numbers over here. The red marker here is the CV cutoff, 20%, and these individual dots correspond to the different population lineages. So this first one is the first donor, second donor, third donor, and so on. Okay, that's the first part of the validation plan. Intra-run precision, same sample, same run. 4.76% readout versus our 20% acceptance criteria. So 100% of populations were below criteria. Now let's turn to inter-run. So same sample, different days, different operators. So if you take this same sample, aliquot, and you have different people run the panel, and then you compare the results, that's what the inter-run analysis helps you identify. So again, trying to find different sources of variability. So here, the average CV across all 3 donors was 6.89%. So if you look again, similar dynamic as we saw before, x-axis is the median size of population, y-axis is the percent CV of the percent of non-granulocytes, and these are all the individual cell types over here from donor 1, donor 2, donor 3. So this is inter-run precision by cell population size. So now we can see the validation results. Intra-run precision and intra-run precision and inter-run precision. CV of 20% acceptance criteria, we notched low single digits, 4.76 and 6.89% respectively. So we consider both of these to be pass in terms of criteria. The other question that we're not going to give a full treatment to is that, is this test comparable to fresh blood? So we have covered that in our previous webinar, which we'll link to as well. We found a great correlation, 0.97% between live and fixed sample processing. This is across all the different immune cell types over here. You can see the x-axis is fresh, the y-axis is fixed. So that's also a great correlation that we're seeing between live and fixed sample processing. And so to sum up, again, the rationale here is why fix blood instead of running fresh is you get this massive reduction in failure rate, 10 to 20-fold reduction potentially. You don't need a physical lab. You don't have all the intersite variability. You can batch all the samples. You can store everything until all time points from a single patient are complete. And then you can dramatically reduce your committed processing costs. You don't need to commit to processing all the samples right off the bat. If your analysis plan, you want to look at a subset of samples, you can do those. You can store at -80 for 24 months until you're ready to decide which samples to analyze. So hopefully this was educational and helped you understand some of the details behind our inter and intra-run comparisons on on fixed blood in Spectral Flow. And if you've got any questions, feel free to ping us or contact us on Takeo.bio. Thanks for signing on.